I was going to give you a water. Actually, let's see. I've made it two bracelets. The other one. Yeah, like what, a water? Yeah. Different. So for a little bit, your dad is coming to those, and that's on the song that I taught. that in the I love him. Come here. No. This is good. Look at it. Transferring money for his sister. No doctor stuff. Right. I got for myself. First time I got 4. Wait, I got one from the back. And then one. Also, from my Mario. We got there early. And the back table tomorrow, and I can work. I have my laptop, and if you want the laptop. You guys got shovels and I have babies. I don't know. I feeding my dog. If you ask for your raw meal, then it would be a lot.
A lot of it.
I don't really, so as a mom, I don't realize what I'm asking for when I ask for that.
You would print it and find it on your phone. I'd be like, $100,000. Like,
I would love to get back on my But you're allowed to ask for it.
it would just come on like a personal hard drive. Something. Maybe a DVD.
Is it okay that I ask for that?
Never gonna make a pay for it. Two nexus got screwed up. They may give you a hard time about it.
And then I was worried, would they give me a hard time and say, oh, we didn't mean to send all of it to you or to the doctor?
Lots of snipe there.
Oh, Dad. Oh. Okay.
Curious how this thing's doing. Checking battery.
Man, you guys look like you're moving in.
I was not for that long. That's what you got.
Taylor misses you, lets you know she got tickets.
This is your, this is from the doctor later. This. These are the things he said. You're positive. Positive for this. And it doesn't mean that thing, it means the word gene, which is your DNA.
Oh, that's how you go. And then see Hao-Fountain.
And so these are the two things. This is the one that we're having a read about. And these are all the doctors. So they're like smart. That's why it puts your brain. Something really smart. Today's the smart. Tomorrow will be one day.
That's why I don't have to listen to it all.
Today's like
Then one the next day.
Yeah. This is one that you and Dad both have. You have a doctor that wants something to say or talk about this one. You got two things. And then we get different. Huh? Oh my gosh. Wanna go find your snack with you? You guys are. What do you want? Your Cheerios?
Yeah.
You wanna take your water? You wanna go to the room? We're gonna start in like 5 minutes. Let me walk you over there. Go put our bowls up. They have a little thing here for the bowl to go.
I'll walk with you.
Here, my sorry, everybody.
But this is like we have in our body, but it's video, like the
Turkey, I know. They basically operated all. Things made it so big.
So Patrick was gonna do Q&A, but he's not comfortable being up front. He's gonna be sitting back there with his mom and dad. But we have, this is information about Patrick, and then these are the same thing. So if you have questions, we put it, we're just giving everybody an advance information to kind of ask him some questions about himself. So, but he's comfortable sitting back there. And just so you know, this is just the Zoom that I'm running a live Zoom. And over there is focusing on the screen so much. I don't know if Tim told you when he moved it over.
Leslie told me she was she made friends with Isaiah. She was dad was
And I kept trying to adjust it, and then it would be like black screen, and then I'm like, so it seems like I'm just gonna move it over.
Okay, everyone, I think we're gonna get started again. So we're gonna start with a Q&A for Patrick. And I'll come over here as close as I can without making the microphones go crazy. Patrick is gonna answer some questions, and there's some information up here about Patrick. And I'll start. If Patrick, would you like to just tell us maybe where you work?
I work with the city of Cary, as the downtown continuum.
Okay, any other questions for Patrick? So what do you do day to day? What is your day look? What do you put on your ballot? Do you keep the park looking really nice that everyone comes through? That's really cool. Okay, other questions?
I just want to ask, I see that a lot, why is that? Oh, okay. Well, it's a great favorite place to live.
I'm Belinda, his mom. I'll give a little background. He was just, we found the Hao-Fountain Syndrome last year through another organization. It's a researcher in California who was doing research on disorders of the corpus callosum. He had given the sample probably 15 years before, and it finally came up that they found something. They found the Hao-Fountain Syndrome.
Well, I'll just say that they found an anomaly. They did not identify the Hao-Fountain. What we had to do, we had to have confirmation testing at Duke University. And that's when they identified the UF7. This was right before the last conference last year, which is why we came to that conference. But Patrick was originally diagnosed with agenesis of corpus callosum, missing the primary band of nerve fibers connected to the brain when he was about 18 months via MRI. And I mean, we knew pretty much right after he was born that, with flow crawling, he had a number of developmental delays. And if you look at the medical part of this sheet on development, you see that a lot of the symptoms that are there are pretty classic for Hao-Fountain, but we never attributed them to Hao-Fountain. We just thought it was connected to his neurological congenital defect. But we've been involved with other organizations, Corpus Callosum National Association, for quite some time. Belinda should have gone to their conferences for a number of years. So we were simply, just never thought about a genetic reason for what was going on. We were just focused on the brain abnormality. And, so that's kind of what brings us here. But again, look at the sheet. Patrick has, again, it's the expectations thing, what are your expectations when your child is a toddler? Patrick is fantastic. He really has. So he's always try to do his best. So I would don't lower your expectations. But I think that mostly your child as an infant and then on, you never know what they're going to be like 10, 15 years down the road. And he's 37 now. It is a lifelong journey, though. That I think is something you can expect as a parent. It's going to be all life, child's entire life. So that's but if y'all have any other questions about anything on there.
Patrick was prepared to answer questions, but I think following the scientists this morning, just like I am, we're all a little nervous. So, so if you have questions, please ask them. But Patrick, I think, does quite well. He loves, it's on there, he loves to swim. He has his own dog. He lives by himself, close to us. And he works full-time for the city of Cary, works part-time for the city of Raleigh. He is a great employee. He has great work ethic, and he loves to stay busy with his two jobs. And also he plays sports with Special Olympics and Unified Special Olympics too. So he's a busy man. On the spot here.
No, not at all. Okay,
It's that one, yeah, just turn.
All right, so I really love dogs. You gotta tell me what's your dog's name, cat dog.
Yeah, speak up loud so you can. Okay, what's your dog's name?
All right, what kind of dog? Black lab. Very nice. Also awesome. Probably mainly for mom and dad. Because this is, looking for their outcome. I saw on here that Patrick graduated from North Carolina. And I'd like to know a lot about that.
The UNC at Greensboro special,
Yes.
beyond academics.
Yes.
There's a lot, I think there was a school that started it in Texas, but I'm not mistaken. There's Clemson University does a 2-year program. NC State just started a new program, 4 years. It's a program for people with intellectual disabilities. They go to college, they have their own classes, but they can also take regular classes with regular students. You are a student there. The first year, you live with whoever you're assigned in the dorm. Depending on your needs, you'll be assigned a helper. He drives, so he did not need to learn the public transportation, and then he's been doing his own laundry since he was probably 12, so he didn't need a lot of extra support in teaching those skills. But it's a 4-year program. I think it was either the second or the third year. He found his own roommates. They rented an apartment. There were 4 of them, and there were 4 guys in the program. I think that was probably one of the best years of his life. They went out, they made plans, and they would go to the movies, and they just had a great time.
Did you do any other research, like any other colleges that you're aware of? Or is this like something, new that colleges are doing or been around for a while? Well, he graduated in started in 2016. By that time, we did do some research. I'm not sure about the technical services. A lot of the programs were 2 years versus 4 years. The reason we decided to go to UNC was, of course, the proximity to where we live. Again, we went there, we visited them, we talked to the folks in the program, what kind of assistance could we expect, what were they going to do for Patrick? And we just thought it would be a really good program. Patrick, she academically struggled throughout. He was always learning by doing physical and visual. And their program is designed, it's not necessarily an academic program as much as a life skill, basic finance, how to advocate for yourself, how to be those individuals with disabilities. And there was a range, as there are, as we've discovered with the agenesis of the corpus callosum. I think with a lot of these disorders, there's a wide range of variants in certain things. But again, the program did really help for dependent, helped him socially. That's, I don't know how else, but again, social skill, being comfortable in certain situations, picking up on social cues that talking to Patrick is very difficult, invasive, or to read nonverbal, what we take for granted. Read somebody and you're like, okay, he's talking up. He just doesn't. That's very difficult. So, but again, this program was fantastic. The only downside was was he graduated in 2020. And COVID hit. And so our plan was, as soon as he graduated, he got moved to semi-independent by himself. And we were going to make that work. And then, of course, COVID, he was with us for a while. But he's in his own town now. And you know, we're fortunate that we can help him out a lot. I mean, I realize that. But he's about a mile away. Why for dinner most days he drops the dog off in the morning, goes to work. His dog comes home and he gets like a shower, comes over, eats dinner with us, picks up his dog. I can tell you, his dog, having an animal, but the dog with dog, maybe something more. But having a companion, emotionally, dog.
I found out about the universities through going to his conferences with the disorders of the corpus callosum. That's what started this whole thing, and then researching and finding one that was an hour away from us. With Patrick, as you can see, he moved before when he graduated from high school, he had moved 12 or 13 times. So education, you start over every year or every 2 years with a nice continuity of education, really kind of made it difficult for Patrick, but it also made him resilient, and he's really nervous, but he can go into any situation. He has started new jobs. He has figured out how to work through that by himself without us there, just because going into new schools every year or every 2 years and new neighborhoods. And so it's kind of a double-edged sword. It was difficult, but he figured it out.
Patrick, it sounds like you've had a lot of different jobs and you're a hard worker. So I'd like to hear from you and maybe your parents about how that worked, like how to figure out what jobs you were going to do and what kind of things you would like to do. Just about that process. I'd like to hear about that.
So what was your favorite job? What led you to do these jobs? Why did you work at the city of Raleigh? Why did you work there? Put him on the spot. Do you like working? Why? Keep it too much better.
You get paid for your job. How do you manage your money? Do you have your own bank account and use a bank card? Do you look at it on your phone too? Is that the best way to track it? Does it like tell you every time you spend money, does it tell you how much you have left?
On Patrick's jobs, he started working when he was 16, just at our commissary on the military base. And then he didn't work for a while because we were moving and he was in high school. But then as soon as we moved to North Carolina, he had just graduated high school in Florida, and we moved to North Carolina, and he worked at the grocery store in our neighborhood. So he could drive 2/10 of a mile maybe on the neighborhood street. And that grocery store closed down. So then he moved to Target. He just likes to be busy. He is not going to sit around. I think, and Patrick, correct me if I'm wrong, he likes working at the Performing Arts Center because he loves, wouldn't know it today, but he loves people. He loves talking to people. And, he'll usher them to their seats and take tickets, and he just loves being around people and talking to them. And with the town of Cary, he was involved in a specialized recreation where they just had movie nights and things like that. And the lady that was in charge of that got him a part-time job with the town and city of Cary, and on a special events crew. So 5 guys, and they go out and they set stuff up if something's gonna happen in a town. And he just loved it. And then he went to a landscaping crew, and then now he's a full-time employee. He has his own truck. He has his route that he does every day. There's probably, 5 or 6 or 7 parks that he goes around and takes care of, and 15, sorry. 15 parks that he takes care of. So, yeah, and he works on the weekend. He has to work 1 weekend a month. After his 40 hours, he works 1 weekend a month, 22 hours on the 2 11-hour days. So, yeah, he loves being busy.
Question. Now, in your state, Patrick, do you have a job coach to help you find a job? No? Okay.
He can, but he doesn't.
And as a job coach, he will help you find a job. Then she'll come in for about a week and then check on her a couple times a month.
Yes.
We were really fortunate, I was in the city of Cary with Judy, who was in the special, who was in charge of the specialized rec program. She was, she knew everybody in the town. She had been there for a number of years, so she's the one who initially got him the job and sort of moved up through the system. But, Belinda would go with him, fill out the application. And they understood that. So we have been fortunate again, especially how willing to work with us and work with Patrick. He's got some great supervisors who understand him, who, make sure understands everything and gets the route on. Again, he's a very visual person, so once he's got his route, done it a few times. He can walk his walk it on. Do those things.
And very dependable. And that's one thing. He knows everyone in the downtown area. He knows all the policemen, he knows the fire chiefs, they all know Patrick.
I would love to ask Patrick what his memories are of high school and if he loved it or didn't love it. We have a kiddo in high school right now, so we hear lots about high school.
Well, he went to 3 separate high schools.
Okay.
So the first one was a homeschool for 9th grade, where we were in Georgia. And then he went to 1 year in Pennsylvania, and then he went to 2 years in Florida. What was the one thing you liked about in Florida on high school?
Or the PC program, leadership program.
Yeah, that's a good program with generally very good students and accepting students. Do you have any advice for moms and dads? Just to love their kids, right? And be supportive.
Hi, Patrick. I am from Puerto Rico and I still live in Puerto Rico. Do you have any memories from there? Anything you like from there?
What did you do before? Want to say about leadership?
How about food? Anything that you like to eat? Like that?
We love Puerto Rico.
We had a good time. We lived on what?
So if you want in the guidance, or guidance,
He went to school at DOE school.
Yeah.
Yeah. But we travel all around, mountain.
We're not that far off from each other. I have that, and I have friends who went to the academy, so that's why I'm.
I have a question for you, Julie. I mean, you said you moved around a lot. That had to be really hard fighting for educational services because I know we were in one town and I spent 10 years fighting that I had to be a lawyer and be a lot of money. I mean, was there a lot of stress with that? I think there had to be. Or was he getting the services he needed?
We were pretty fortunate. Most of the time we were in Department of Defense schools on a
Okay.
military base, so, and it just kind of carried over. It's always, he doesn't, present well with new people, but once you get to know him, you know, they're always going to test in the beginning. I'm like, well, you got to test him 6 months after you know him and it's a different person. The other thing, he has agenus of the corpus callosum. It is literally missing part of your brain. It is concrete where school systems can understand that. You know, sometimes autism, I mean, everybody kind of understands it, but everyone's different. Everyone's different with ACC too, but it was something that they knew it was concrete in the brain. They knew there was an issue. So we never had a problem getting services at all. OT, PT, speech, yeah.
We had a lot of fun.
If this goes to the diagnosis, so he's always had a diagnosis, or he's always had something, this congenital, brain defect that, you know, doctors can look at and clinicians and providers, and schools, et cetera. But I will tell you, it might be downplayed, she fought in a lot of, we were in a number of different schools where she had to go in, talk to them about the IEP, and what was right for happening, what was the correct setting, should he be mainstream? Should he be pulled out? Should he be pulled out part of the time? Should he have somebody in the classroom with him? So she was in school all the time. She volunteered just about, and we were fortunate here again, I was in the military. She stayed at home, and not at home, she was out doing something all the time, but she had the time to spend in the schools, and we had the building. So she was always in the schools, always advocating for that.
I was in school so long.
Yes.
I'm glad
So,
those days are over.
you know, that made a big difference, having her there and advocating for them, making sure that you have the services that we want.
And they were good about that because with us, they,
Yeah, and again,
it's always about the money.
I think, depending on where you're at, DOD
What
schools,
district you're at?
DOD schools, Department of Defense schools on post.
Federally funded.
Generally better. Now, we also live in Northern Virginia schools that were very good. It's kind of where.
Florida, probably Florida. Oh, Pennsylvania, Patrick just said, yeah, I think he had some issues with the schools in Pennsylvania. The school he went to in Florida was the largest high school at the time in the country, 5,700 students in one high school. So that, yeah, that was a little challenging.
He's doing great now. You're doing wonderful.
Well, if I want to expect Patrick, why don't you start driving? What age did you about?
He started driver's ed in Pennsylvania, so he was 17, because he repeated kindergarten in Germany. And then he did driver's ed again in Florida. So he got his license when he was 18 years old. Yeah.
And I worked with them all out too, in Florida, drivers ed. And I know, you know, again, don't lower your expectations, but driving is a very difficult skill for not just, you know, so you have 7 to difficult skill for what do we call them, normal people.
Yeah.
I mean, that's a difficult skill, and he started off driving just locally on the back streets, and just, and primarily drives places where he's clear, stuff like that. So that's, that can be challenging for anybody. If you have children and you take them through the driver's license, and you have 3 other children, and you're taking those children through the driver's license.
With driving, one of the things that helped him, like when he went to school to the University of North Carolina in Greensboro, we took him a couple times and he wanted his truck there. So then he followed us a couple times, and then it would be, he was on the phone on his car and we were talking to him the whole time he was there and driving there and back, because it was about an hour and 10 minutes drive. And then he did everything on his own. And one time coming home, we didn't find this out till later, he missed the turn and, you know, he's driving around with his GPS and about after an hour and a half, he figures out how to get home. But he did it all on his own. He didn't call us. He didn't tell us anything. He just figured out how to get home. So
And that's it, technology. I mean, when you, the youngster, I see, Adam is already, you know, on the phone, and we didn't have any of that. But, you know, now the phone, he can call, text, so always go to the reason what the right now through the phone, texting, learn all of that. Better on the time, you know, the laptops, some of the people love to look at YouTube videos and they're all about everything. Learn so much now through not having to read it, just by looking at it, listening to the people. Technology.
So, and then we need to get on with the program. But please come up and ask your questions and discuss anything.
And
Okay.
especially tomorrow, we're gonna have a parent panel,
Okay.
so yeah, it'll be a great time to exchange. But yes, thank you so much, Patrick, for sharing. All right, next up is Christian Schaaf, and he is going to be talking about their latest research.
Okay, thank you. Thanks for having me. I'm gonna try and give like an overview of the clinical research and what we've learned over the past 10 years, because it has been 10 years since the first manuscript was published, and I feel that I've been kind of walking this walk with many of you for quite some while. This is a picture of our first USP7 conference in Houston in 2017, as we walked our way into the seminar room. Looking back, it's amazing to see how far you all have come, how far your kids have come, but also to see how far the group has come and the organization, because, you know, 8 years ago, it seemed almost impossible to have such a dedicated group of researchers and physicians together, all working and all interested in advancing the field of USP7 research. Okay, you all know that USP7 mutations cause Hao-Fountain Syndrome, but like, what are the underlying mechanisms? One mechanism that I think is very well established is the mechanism of what we call haploinsufficiency. So lacking one copy of the USP7 gene is sufficient to cause Hao-Fountain syndrome. We're not aware of any single person who lacks both copies of the USP7 gene, because we feel that probably that's incompatible with life. But lacking one copy or lacking the function of one of the two copies can cause Hao-Fountain syndrome. Now, as often in life and certainly in medicine, things are a little more complicated, and Irina and others have kind of generated evidence that maybe also having too much of USP7 or having too much activity of the USP7 protein may also not be ideal. And Claudia, who has been with us last summer in Heidelberg and who's continued her research on USP7 duplications, will also present kind of some data in that regard. So I want to kind of briefly summarize what we've learned over the years. As mentioned, that first publication came out in 2015 by Hao and Fountain and others, which is also the reason why the condition is called Hao-Fountain Syndrome, because the organization OMIM that kind of keeps track of all the genetic disorders and also is charged with naming rare genetic disorders typically uses the names of the initial people who first reported about a gene-disease relationship and kind of uses those names to then kind of generate a disease name. So at that time, this was a very basic science oriented publication by the lab of Brian Potts at UT Southwestern at the time. That identified the protein interaction networks and the functions of USP7 as it relates to TRIM27 that was already mentioned earlier today, and MAGEL2 being a gene and protein that my lab had extensively been studying. And they kind of define this protein complex that's now called the MUST complex, of USP7, TRIM27, and MAGEL2 that fulfills a lot of the functions that relate to protein recycling, ubiquitination, and deubiquitination that was mentioned before. Now, we knew that if you lose MAGEL2's function, that causes a neurodevelopmental disorder, which is called Hao-Fountain syndrome. And Brian and his team showed that within this complex, no matter what you take out, whether you take out MAGEL2 or whether you take out USP7 or whether you take out TRIM27, the molecular and functional consequences are pretty much the same. So we speculated at the time that, well, if you take one partner out and it causes a neurodevelopmental disorder, maybe lacking one of the other partners would also cause a neurodevelopmental disorder. And so we went through our databases of individuals who had undergone exome or genome sequencing and had kind of high-level chromosome analyses, and sure enough, identified at that time 7 individuals with USP7 mutations. 6 of them were individuals who were lacking one copy of the USP7 gene, and 1 had a point mutation that kind of disrupted the function of the gene, and that led to that initial publication, and those were the first 7 individuals to be reported in the literature. Much more clinical information was then gathered in over the next 4 or 5 years, and Mike Fountain, who was one of the PhD students in my lab at Baylor College of Medicine at the time, put together this publication, which is kind of the first clinically oriented study on Hao-Fountain syndrome that reported 23 individuals, the initial 7 and then 16 new individuals, and kind of more thoroughly describe the clinical features that we see in your kiddos. So the main features that were reported were developmental delay, behavioral characteristics, in particular affecting speech, but also autism spectrum disorder or autistic features, ADHD, self-hurting behaviors, and some feeding difficulties, hypotonia, low muscular tone, kind of leading to those feeding difficulties probably, but also kind of the inability to coordinate the complex mechanisms that are needed to swallow, subsequently failure to thrive, especially in the early years. Now we know that right after birth, in the first couple of years, kids with Hao-Fountain syndrome tend to have kind of a lower weight and kind of rather short stature, but then as they get older, they seem to catch up quite well. And actually, the older kids with Hao-Fountain syndrome tend to be in the higher percentiles for kind of weight and length. Mike also kind of discussed eye anomalies with a very high prevalence, in part related to the hypotonia again, especially with crossed eyes, like the inability to focus very well, which then can cause nystagmus, and also highlighted the abnormal MRI findings with the abnormalities, especially of the white matter, including the corpus callosum and other structures in the brain. And then in 2019, I moved back to Germany, and I now direct a genetics institute at the University of Heidelberg. And one of my students there, Moritz Wimmer, kind of followed up and did another questionnaire-based study reporting 32 novel patients with Hao-Fountain syndrome, and kind of discussed their clinical manifestations. And I think like some of the main outcomes of that study were that gender, age, and the mutation type, so whether it's a deletion or missense mutation or nonsense mutation, don't really influence the severity of Hao-Fountain syndrome, but missense variants in the catalytic domain, which is one of the main structural domains of the USP7 protein, do affect the severity as they do cause a more severe phenotype. And you can see that on the far right in that panel. So the bar that's marked with CD, which stands for catalytic domain, has a higher severity score, which means more severe clinical manifestations compared to missense variants in the other that affect the other protein domains. And the establishment of that kind of severity score was also something that came out of Moritz's study at the time. So in Heidelberg, we are particularly interested in ultra-rare neurodevelopmental conditions, and we do keep registries of multiple conditions, including Sharp-Yang and some other conditions that we've discovered over the years. Hao-Fountain is one of them. And on the bottom, you see the information that we keep in our registry of the patients who agree to enroll. So we keep some personal information, genetic information, which mutation, how does that affect the protein? We keep clinical information. We keep track of biomaterials that are available. So on which individuals do we have fibroblasts, on whom did we reprogram induced pluripotent stem cells and others. We then further on kind of share those biomaterials also so that they can be kept in a biorepository also in the United States. Which other studies they participated in. We keep photos and consent documents and also contact details from the referring physician and the family, so that if new kind of studies come up, if we are approached by other researchers around the world, or if we have new ideas, we also use the registry to recontact the families so that they can participate in those subsequent studies. To this date, in our registry, we have information on 235 individuals with USP7 variants. On about 50% of them, we know exactly the inheritance pattern, whether it's a de novo mutation, whether it was inherited from an affected parent, whether it was like germ cell mosaicism or others. We can go into that. We have personal details of gender and age in 66% of those. We have the contact information of the family for 76% of cases. About half, we have the contact information of the physician, and a quarter we have information on both of those. You can see on the right that we have equal numbers of males and females in our registry. And on the top, you can see that 40% of patients in our registry come from the United States, 47% come from Europe, only 4% from Southern America, 5% from Canada, and 4% from other countries, including Africa and Asia. You can see the distribution of the age group here. More and more young individuals are diagnosed because exome and genome sequencing are now applied on children with hypotonia, feeding difficulties early on. But also slowly, but really slowly, we're getting information on older individuals. This is much harder because a lot of the individuals who are Patrick's age or older, the families have kind of closed that chapter of kind of trying to look into what the underlying cause might be, and also because they not having the regular physician visits like the well child visits, you know, where you have to go to the doctor on a regular basis. They not refer to genetics anymore. But like in our database, as you can see, we have a pretty good distribution, you know, with kind of a main focus on individuals who are in their kind of school age years, I would say. But 30% in our database are actually 17 years or older. What's the distribution of variants? This is also kind of a nice spread. You can see in orange, the majority of cases are missense variants now. This is different from the early studies. Initially, we found a lot of kind of deletion cases, but with the advent of genome sequencing and exome sequencing, we now have more and more missense variants in our database. For 50% of the cases, we know the inheritance pattern. Out of those, 86% are de novo, which means the parents do not carry that variant. Particularly interesting are certainly the 14 inherited cases where we have a parent who carries the variant and passes it on to his child. Obviously, each and every person who has a USP7 mutation has a 50% chance of passing that on to each and every one of his children. These are inherited cases where affected individuals pass it on to their children. And then we have 2 individuals in our database where we suspect germline mosaicism. That means neither one of the parents carries the variant, but still the family had multiple affected children with the condition. And this is due to a situation where multiple cells in the germline, either in the production of sperm or in the production of egg cells, carry the mutation. And there is always that remaining chance, even though you cannot find the variant in the blood of the parent. So this is part of our genetic counseling as well, and does not only apply to Hao-Fountain syndrome, but all conditions that follow autosomal dominant inheritance. We always counsel that there's a typically say 2% or less chance of having another child, even though the mutation was not found in either parent's blood. Okay, and then over the past years, we conducted this clinical research study, which was the first prospective clinical research study and the first kind of structured clinical research study for USP7 related diseases, supported through the fundraising of the USP7 Foundation. And so what did we do? We invited affected individuals to Heidelberg University Hospital. This was a 3-day visit, and the goal was to characterize kind of the metabolic and hormonal phenotype of Hao-Fountain syndrome, to look at brain function and brain structure, to do lumbar punctures and collect cerebrospinal fluid and investigate the neurotransmitters in the CSF, and also do some skeletal phenotyping. And I'm most grateful not only for the funding of the study, but also to the individuals who kind of went through all the efforts to travel to Heidelberg and participate in the study. This research study also made contributions to other projects. Biosamples were sent to Lieselot and the team in Amsterdam to participate in the EpiScience study. We collected biomaterial that was deposited in the biobank for the USP7 Foundation. And we took photographs, the facial photographs that were shared with the Gestalt Measure database, which is a artificial intelligence system to recognize facial features and predict the most likely genetic alterations that underlie the disease of that person who was photographed. which could, and actually in clinical practice is already a very important and very inexpensive tool to also help identify the underlying cause of individuals with neurodevelopmental disorders. So 11 individuals with Hao-Fountain syndrome traveled to Heidelberg to participate in the study. You can see that we were somewhat biased towards females. 9 of the 11 individuals were female individuals with Hao-Fountain syndrome. The average age was 11 years old. And you do see that we have a number of different genetic alterations represented, but majority of cases being missense variants, with 7 out of the 11 cases being missense variants. When you look at the body measurements of those patients, you can see what I told you before. So at birth, you can see body weight in blue, body height in red, head circumference in green. You see head circumference at the time of birth is average, kind of represents the normal range of the population, but body weight and body height on average are below the 50th percentile. When you look at the time of enrollment in our study, when, like I said, the average age was 11 years old, this has changed. At that point, body height is right at the 50th percentile on average, but body weight has kind of moved up and is now above the average in the general population, as is head circumference and BMI. So the average BMI of the study participants was around 19. What were the main clinical features in that cohort? So you can see that each and every one had a history of developmental delay and or intellectual disability. We had 91%, meaning 10 out of the 11 participants manifest autistic features, but only 55% actually meeting bona fide criteria on autism spectrum disorder diagnosis. Interestingly, 25%, so a quarter of all the participants in the study, were on growth hormone therapy. This is something that kind of goes back to the relationship of MAGEL2 and USP7, because we know a lot about growth hormone therapy and the benefits of recombinant growth hormone in Prader-Willi syndrome and Sharp-Young syndrome. So it has been suggested also that growth hormone may be beneficial in Hao-Fountain syndrome, not necessarily for longitudinal growth, but more so for shifting adipose tissue to lean muscle mass and increasing kind of the buildup of skeletal muscle, which then can help with the achievement of certain motor developmental milestones. You can see that 67% had a history of neonatal hypotonia, about half of them had a history of seizures. Gastrointestinal problems, in particular constipation, are very frequent among individuals with Hao-Fountain syndrome. Eye anomalies, again, very high prevalence. Sleep difficulties we heard about before, related to Larry's work, about half of them, and this also reflects kind of the percentage that's reported in the literature. Actual sleep apnea in 1 out of 5, and something that was new to us that we had not been aware of, the problems that affect the skin or the hair. So we seem to have a fairly high prevalence of eczema or psoriasis among individuals with Hao-Fountain syndrome, and also what's been mentioned at least twice, I think, today, hypertrichomania, which means the pulling of hair, exactly, also is something that is mentioned again and again. Have to practice that word. In the neurological examination, and there was a standardized neurological examination by our pediatric neurologist at Heidelberg Children's Hospital, we found impaired reflexes in 11%, 55% had impaired gross motor skills or gaze disturbance, 70% had impaired fine motor skills. So this is something that I wasn't aware of before. The prevalence of fine motor impairment seems to be higher than the level of gross motor impairment. Nobody had ataxia, and hypotonia at the time of examination was only present in 11% of the individuals. This is particularly interesting because when we asked parents about hypotonia, it was reported in 86% of cases, maybe also biased by our own kind of publications and me asking over and over again whether your children have hypotonia. But like based on what our like expert neurologists say is that at the age of enrollment in the study, only 11% had actual hypotonia, which to me indicates that a lot of your kids have neonatal and infantile hypotonia, but the muscle tone actually improves over time. And by the time they kind of school age, teenage kids, most of them probably do not have hypotonia anymore. Okay, we also looked into kind of hormonal function and metabolic function, and we did glucose tolerance tests. So we looked at fasting glucose levels, and you can see that 3 out of the 11 participants had fasting glucose levels that were above the normal range. And then we fed a kind of sucrose solution to the participants, and 2 hours later measured glucose levels again. And you can see that 5 out of the 11 individuals had glucose levels that were at least in the pre-diabetic range. In fact, 2 out of the 11 had glucose levels clearly in the diabetic range. So this is something that I think will be important for all of you to keep in mind in regards of anticipatory guidance, to ask your providers to look at the possibility of your child developing diabetes or pre-diabetic state, and if so, to intervene early. We did standardized photographs and looked at facial features, and you can see, I mean, this is a very long list of very detailed kind of physical facial features on the right side, but you can see that there are certain facial features that appear again and again, and there is some kind of, I wouldn't say like for the average geneticist, this is not a condition that we recognize. So it's not a condition where the patient walks in and we say, this is Hao-Fountain syndrome. I've seen this 5 years ago. But there are facial features that do appear again and again. And as so often in life, I guess artificial intelligence is so much better than us. So artificial intelligence can recognize the facial features of Hao-Fountain syndrome. And this is indicated on the right side. The similarity of the facial phenotypes in Hao-Fountain syndrome is much more similar than what you would expect by random distribution among individuals with a neurodevelopmental disorder. And this is important because, like, with those photographs being submitted to the Gestalt Matcher database, it can help future physicians recognize and diagnose Hao-Fountain syndrome earlier. We did talk about brain abnormalities, and so the individuals who participated in the study also underwent sedation and brain MRI analysis. And you can see that MRI findings are quite frequent in this patient population. Particularly affecting the white matter. So deficits in white matter are a fairly common effect. More than half of the patients, we do see reduced volume of the corpus callosum or absence of the corpus callosum in a certain percentage. And we can also have structural abnormalities. In 3 out of the participating patients, we did see structural abnormalities, such as these formations that can affect the white matter, but also other structures like arachnoid cysts on the left-hand side, or cerebellar hypoplasia on the right side. It's particularly interesting because the cerebellum kind of coordinates our motor movements, like impulse, like one of the jobs of the cerebellum. And despite the fact that this individual had quite remarkable cerebellar hypoplasia, there was no ataxia on a neurological exam, which you would typically expect when you see that level of hypoplasia. And then we did skeletal phenotyping, so X-ray studies. 36% of patients with Hao-Fountain syndrome in our cohort manifest scoliosis, and we looked at bone health. So why did we look at bone health? Again, you know, this is because of the relationship to Sharp-Young syndrome and Prader-Willi syndrome. And we know from Sharp-Young that bone mineral density tends to be decreased among those individuals. So we wanted to find out if that was also the case in Hao-Fountain syndrome. And we did find a decreased bone health index in 60% of the participants in our study, with 40% being in the range of osteopenia, which means the score, like the level of bone mineral density, is decreased, but it's not quite at the level of osteoporosis yet. But 20% of the patients actually had manifest osteoporosis with a T-score of -2.5 or lower. Again, something that's important to keep in mind. It's not easy to determine that bone mineral density because you would have to go through a DEXA scan, and like DEXA scans, if at all, they only done on kids 5 years or older. If the kids are younger than 5 years, you can determine bone mineral density based on hand X-rays. But you can take kind of small measures, like even if you don't know the actual bone mineral density, by making sure that the calcium and vitamin D levels are in the normal range or the upper normal range. And then especially for the older kids, teenagers, young adults, older adults, I think it's important to get DEXA scans and really determine what the bone mineral density is like. This is a bad one? Okay, especially when they're older, you want to find out because if they have osteoporosis and it goes unrecognized, obviously they at a much increased risk of having fractures when they hurt themselves, when they fall, etc. Okay, and then I briefly wanted to touch also on ongoing research. So it seemed striking from our database that there were multiple individuals with congenital abnormalities of the kidneys and the urinary system. So we do have an ongoing questionnaire-based study that Felix Funke, a medical student from Heidelberg, is conducting with us to look into that. The acronym is called CACUT, which stands for Congenital Anomalies of the Kidney and Urinary Tract. This is still in the early stages, but there are two published cases of Hao-Fountain syndrome who have congenital kidney disease, and then we have collected another 5 individuals where we see kind of structural abnormalities of the kidneys and the urinary tract. So this is something that we have to follow up on. And we would also be interested in whether any researchers who are working on animal models, whether this has been seen or looked at. Oftentimes, also in the animal models, we only see what we look at. So like the mouse and rat models for other diseases that we study, I've never looked at the kidneys because, you know, my primary focus is on the brain. So sometimes it helps to kind of push people into that direction to kind of start looking, because there may be a relationship. And also, I would say for every child with Hao-Fountain who has never had a kidney ultrasound, I would recommend getting a kidney ultrasound. Okay, so in summary, I feel that some of the most relevant features include speech delay. I think speech delay by itself is the most prevalent feature of Hao-Fountain syndrome that we're aware of. Hao-Fountain patients share a high degree of clinical similarity, even though the spectrum is really wide, but also share a high degree of facial phenotypic similarity. And when we look deeper into the body, we can see certain patterns arising, including certain abnormalities of the brain, in particular related to the white matter. Some of the things that I mentioned will have implications as it comes to anticipatory guidance and surveillance, including evaluations for scoliosis, osteoporosis, pre-diabetic state, and kidney structure and function. And I just wanted to briefly mention this because we've been working on this over the last half year. One of the databases that a lot of physicians rely on as it relates to congenital genetic disease is gene reviews, which is kind of the expert database for rare genetic disorders, kind of hosted in the National Library of Medicine. And we've applied and we've been working towards getting a gene reviews chapter for Hao-Fountain Syndrome. And I was hoping that it would be published by today so that I can kind of present it to you. And I contacted the editors at University of Washington, but they said it's still in the works, but it has already gone through 2 rounds of review. So I expect within the next couple of months, the gene reviews chapter on Hao-Fountain Syndrome will be published, which then will be like an incredibly important resource to physicians and genetic counselors worldwide, as it will present like an up-to-date summary of the clinical features, the genetic features of Hao-Fountain, but also give recommendations that relate to management, anticipatory guidance, and surveillance, including some of the things that I mentioned today. So with that, I'm finished, and I wanted to thank you all for allowing me to be part of this journey, and I look forward to what else we can accomplish in the next 10 years. Thank you. I think we'll skip questions because we're kind of running a little late, and give Claudia a chance to present the data on the USP7 duplications, and then maybe we can go through questions after. And like tomorrow, I'm also part of the family conference, and basically will present very similarly the data that I showed you today. So kind of individual questions about like my child has this, so that we can probably kind of go much deeper tomorrow.
First of all, I would like to thank everyone here for the invitation. I am a physician doing my first year of residency, and I will give you some clinical insight about 10 subjects with USP7 duplication. First of all, as we know, Hao-Fountain is a rare neurodevelopmental disorder. We know that the neurodevelopmental disorders are defined in DSM-5 as a condition that develops during the developmental period, leading to some deficits that affect the personal, the academic, and the occupational functioning. We know that the main feature of Hao-Fountain are clinical involved cognition, behavior, and motor development, but there are also some systemic features such as GERD or hypogonadism and other features. We know that Hao-Fountain syndrome is caused by USP7 mutation. In particular, we have point mutation and deletion, but we were also asking to ourselves if also a duplication could cause a phenotype. If so, USP7 could be considered as a dosage-sensitive gene, which means that too little or too much of its product can lead to a certain phenotype. We have ClinGen that provide us some scores of uptake-sufficiency and triple sensitivity. In particular, uptake-sufficiency is when one copy is not enough, so the lesions are pathogenic. And triple sensitivity is when one extra copy can be sufficient to cause a disease. In this slide, I will just show you some example of other dosage-sensitive genes where both deletion and duplication lead to a certain phenotype. I would like to show you that sometimes the problem is not the gene, but rather the imbalance of their entire locus that causes phenotype. There are some evidences in literature on USP7 duplication. In particular, we have some mice models, and also we have some clinical case reports. And lately, Shanta et al. Described the relationship between USP7 duplication and ASD. We conducted a recruitment campaign via family, physician, and online database such as Decipher. We were able to reach out 12 patients, but then we had to exclude 2 of them. One, because we find another pathogenic variant in FOXP1 that could overlap the phenotype, and the other one because the data were not reliable. They were not complete. So we asked for array CGH reports. Our inclusion criteria was a duplication smaller than 5 MB, including USP7, but as you can see, we have a very huge variability in our sample. We have the largest duplication that is 4.1 MB encompassing 30 genes, and the smallest one that is just 36 kb and partially containing USP7. Most of these duplications are inherited, and when it was possible, we asked for the parents to participate to our questionnaire. In particular, as you can see, we have patients 6 and 7, which are daughter and mother, and also we have patients 9 and 10, which are son and mother. We also, patient 5 inherited this duplication from his father, who was not included in our questionnaire, but we know for sure that he has a formal diagnosis of autism spectrum disorder. And patient 4 inherited this duplication from her mother, but we were able to enroll her. And also patient 2 inherited this duplication, but the father was one of the patients that was excluded from our analysis. Then we have patient 1 and patient 3, which present a de novo mutation, and we don't know the inheritance of patient 8. So we made a standardized clinical questionnaire comprehensive of birth and developmental history, physical characteristics, neurological characteristics, and when it was possible, we also asked for some clinical reports to make a better medical assessment, medical context for each case. Then we were able to collect 4 blood samples to perform the DNA methylation signature with the epsign assay that Lizelot previously exposed. And then we asked for the Gestalt Matcher team to see if there were some similitudes among the facial phenotype of these patients. In particular, we received 5 pictures coming from 4 patients, and they performed an analysis thanks to the AI, and they made a comparison among our sample, some controls, and also with patients with Hao-Fountain Syndrome. Now we'll briefly show you some results from the questionnaire. In particular, all the patients were delivered at term with normal length and weight for gestational age. Some of them reported hypotonia, neonatal pulse-up, and feeding difficulty. We also asked for developmental milestone. It was registered a speech delay and motor delay, and we also asked for other neurological signs such as hypotonia, hyposthenia, and motor skills impairment, communication impairment, and other signs. There was a big prevalence of autistic traits, but only 2 of them received a formal diagnosis. Other 2 of them weren't able to answer specifically to our questions, but they showed an alterated behavioral profile. And as I said before, one parent of this patient that shared the same duplication as a formal diagnosis of ASD.
There were a lot of people also showing attention deficit and hyperactivity, but only 3 of them had the official diagnosis. The other never got an official test assessment. And finally, it wasn't possible to determine the IQ range because most of them didn't provide us with official test results. And in this slide, I just highlighted the most common feature resulting from our test, but I would say once again that most of them didn't receive a formal clinical evaluation. So in this slide, I would like to show you that it's not possible to talk about a unique phenotype, but rather a spectrum of clinical manifestation. And I think it was very important for us to collect this data from the families because this allowed us to understand not only the medical part, but also how much this condition is affecting their daily life. Because in rare disease such as this, I think that the alliance between families and doctors is fundamental. And here I would like to show you that, as I told you, the 16p duplication is like a border spectrum, but if we can say that USP7 is the critical gene for this condition, we can see that the phenotype is more severe when there is the duplication of other genes, such as ABAT or PMM2 or TMEM186. So the relation between the duplication size and the phenotype is just partial. I mean, it's true that the larger the duplication it is, the more severe is the phenotype, but actually it seems to be more relevant the critical gene content. And finally, I will show you just one slide about the episignature because, as I said, we have a very small sample. But what is very interesting about this analysis is that 2 patients present a very opposite methylation profile in comparison with people with Hao-Fountain syndrome. And in this chart, I will show you the facial analysis. In particular, this chart shows you how much patients in our group look alike. In particular, the darker squares mean a strong facial similarities. As we can see, there are some patients that are very similar to each other, and it's also interesting to see that this machine, this AI, was able to recognize the same patient at different age as similar. In particular, we have 2 charts. The first one represents the comparison among our cohort and people with USP7 syndrome. The second one, just the cohort of USP7 duplication. We have 3 curves. The blue one, which is represented by people with the same syndrome, so it's a control for similarity. Then we have the red one, which is represented by people with different conditions. And then we have the orange one, which is represented by people with Hao-Fountain syndrome and USP7 duplication. As we can see, the orange curve is on the right side after the threshold, which means that there is no facial similarity among this cohort. But if we look at the second graph, we can see that the orange curves, which is represented only by patients with duplication, is on the left side before the threshold, which means that there is a similar facial appearance. So the machine was able to recognize similarities among these carriers. And this slide is just to say that it's possible that the same duplication looks different among the carriers. And the key message is that the duplication is not deterministic on its own, but the phenotypic impact depends on other genetic and environmental context. And finally, I will discuss about the limitation because I know that our study has some limitations. For example, we have a very small cohort size and a wide variability in the duplication side. Then there is the lack of the clinical assessment and the main investigation process was made by the questionnaire. But I think it's important to also overcome these limits in other studies, like using a bigger cohort, or also a more homogeneous data set, using a standardized multidisciplinary clinical evaluation, and also an instrumental assessment. Finally, there is no clear correlation between USP7 duplication and a unique phenotype, and there are no similitudes between Hao-Fountain and our cohort. But I think these findings are very important as a starting point, and to highlight the potential biological and clinical relevance of USP7 duplication, and hopefully there will be more clinical studies about that. Thank you for your attention.
Questions for Claudia?
I was gonna say, if not, we're gonna do like an open session, do we wanna have lunch, because lunch is out there, do we wanna do lunch first?
I think we can already smell lunch. So I would suggest we go grab lunch. If you guys have any questions for Claudia or myself, feel free to come up to us, and then like want to kind of start again at like 10:01.
Yeah, that sounds good.
Thank you.
1:10. All right, and everybody help yourself to lunch. If it does smell, I can smell it. It smells good. Yeah.
Look one that. Yeah.
She's very, I was trying to tell you that. Missy Burns. Sometimes it's.
How old? 11, mine is 8. And I was here. Yeah, no, you're fine. If you wanna find me. Some other parents, very helpful. When I was first meeting in 2019, there's not maybe all my team adults here, like a lot. Some people want to do lots of like
I'm not sure. I mean, like, I can't really tell.
Yeah. I was thinking about I got you. The big small, right here. Yeah. What happened?